Hello, in this video we're going to discuss mitosis and meiosis and compare them side by side. Both of these processes are involved in the cell division, which itself is a part of the cell cycle. Let's take us, humans, as an example for this. Our cells can be divided into two major groups, somatic and germ cells, with a simple difference that germ cells are the cells involved in our reproduction. So these are sperm and egg cells. And somatic cells include all the rest. So this can be any cell of our bodies. Somatic cells in our bodies, as well as in bodies of any other sexually reproducing organisms, are typically diploid, meaning that they have a pair of each chromosome. While germ cells, which are haploid, only have a single chromosome of each type. So for us humans, it will be 23 chromosomes in a haploid germ cell and 23 pairs, so 46 chromosomes in total, in a diploid cell. But why does all of this even matter for us now? Well, you see, the first difference between mitosis and meiosis that we can define here is that somatic cells use mitosis for the cell division, whereas germ cells primarily use meiosis for this purpose. And it is not a coincidence, because in the process of meiosis, the number of cell chromosomes gets reduced by half, which is not optimal for the somatic cells that can undergo up to 60 cell divisions during their lifetime. That is why they divide using mitosis that doesn't affect the number of chromosomes. However, a sperm or an egg cell needs to be produced once to serve its purpose, which is to merge with another germ cell to eventually produce another diploid organism. Both mitosis and meiosis are a part of a larger process called cell cycle, which consists of two main phases, interphase and M phase. The main function of the interphase is to prepare the cell for division, and it consists of three steps, G1, S, and G2 phase. G1 and G2 phases are dedicated to the growth of the cell, which makes sense as the cell needs to grow before it divides. But in the S phase, there are two things that happen and that are important for us to remember. The first one is centrosome duplication. Later I'll explain what a centrosome is, but now let's focus on another thing that happens during the S phase, which is chromosome replication. You see, we cannot just simply get two identical chromosomes from the single one. That is why it needs to replicate first. After this process, every chromosome in the cell will consist of two identical halves, connected by a centromere. These parts are called sister chromatids. But wait, this is a chromosome and this is a chromosome, right? Right, a chromosome can consist of either one or two chromatids, and we use C to represent the number of chromatids in the cell. Let's begin with the mitotic cell cycle. To simplify the explanation, we'll take a diploid cell with a haploid number of chromosomes n equal to 2. As the cell is diploid, we'll have 4 chromosomes and 4 chromatids in total in the beginning of the cell cycle. After the growth in the G1 phase, it will reach the S phase, where the chromosomes will replicate and the cell will have double the number of chromatids. After that, we have the G2 phase followed by the M phase. The M phase is the mitosis itself, and it consists of 4 phases: prophase, metaphase, anaphase, and telophase. In the early prophase, the chromosomes of our cell start to condense, and centrosomes that replicate in the S phase begin to move to the opposite poles of the cell by forming the mitotic spindle. In the late prophase, some people also call it prometaphase, nuclear envelope starts to break down, which allows microtubules to attach to the chromosomes. In the next phase, called metaphase, when all chromosomes are captured by the microtubules, the mitotic spindle starts to align them in the middle of the cell. This place is also called metaphase plate. The separation begins in anaphase, when sister chromatids separate from each other and then are pulled to the opposite poles of the cell by the microtubules. Microtubules that didn't attach to the chromosomes push against each other, thus making the cell longer. In the telophase, which is the final phase of the mitotic cell cycle, we can already see two identical daughter cells that are not separated. In this phase, both of the cells start to re-establish their initial form. Each of them forms a nuclear envelope around the chromosomes, the spindle disappears, and chromosomes start to decondense, and so they return into a thread-like form. At the end of the telophase, cells are finally divided in the process called cytokinesis. Moving on to meiosis. Meiosis is pretty similar to mitosis, but the first major difference that we're going to notice here is that meiosis actually consists of two stages instead of one in mitosis. These stages are called meiosis 1 and meiosis 2. Before entering meiosis 1, a cell must go through interphase. As in mitosis, the cell grows during G1 phase, copies all of its chromosomes during S phase, and prepares for division during G2 phase. During prophase 1, chromosomes condense, as in mitosis, but they also pair up in order to trade their parts in the process called crossing over. The pairs of connected chromosomes are called bivalents, and the chromosomes are called homologous chromosomes. Crossing over occurs for a reason. It allows the formation of new, unique, and remixed chromosomes, which leads to genetic variability. After prophase, similar to mitosis, spindle begins to attach to bivalents and lines them up at the metaphase plate. In anaphase 1, the homologous chromosomes are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart. Finally, in telophase 1, chromosomes arrive at opposite poles of the cell. Cytokinesis occurs, and now we have two homologous daughter cells. Now, when our cells move from meiosis 1 to meiosis 2, no replication of chromosomes occurs during the interphase between these two phases. So the number of chromosomes, as well as the number of chromatids, stays the same. Meiosis 2 itself is similar to mitosis, with the only difference that the cells are haploid from the beginning. So if we compare our example cell with the somatic cell in the beginning of mitosis, we'll see that it has half as many chromosomes. As I said, meiosis 2 is similar to mitosis, so the number of chromosomes after meiosis 2 will stay the same, while the number of chromatids will get reduced by half. In conclusion, let's summarize the differences between mitosis and meiosis. That is all for today. Thanks for watching, subscribe, and I'll see you in the next one.